Sorry. Pedro Lowenstein is up. Pedro, he's going to talk about in the theme of brain cancer.
All right, thank you very much for your kind introduction, and thank you to the organizers of the conference for putting this wonderful conference together and the invitation to come and talk to you about some of the most recent work that we've been doing on the large-scale spatio-temporal organization of gliomas. The message I will be giving you today is that we believe that the gliomas are actually organized as liquid crystals, and I'm going to go into the details on why we say that and what the consequences are. So this is a collaboration between us here at the University of Michigan with Sebastian Moch from Arizona State here in Tempe, Tomás Grijera from the University of La Plata in Argentina, and Carles Blanc Mercader, who is at the Institute Curie in Paris. So let's go quickly to, there we go, what liquid crystals are. So when we all were in school, we were taught that there were three main phases of matter: solid, liquid, and gas. Turns out there are several more, and a particularly important one is this one here called liquid crystal, which is literally in between solid, so it has some characteristics of a solid, and a liquid. So in a solid, you have orientation and periodicity of the molecules. They're somewhere in space, and they're fixed in that space. While in liquid crystals, you have an orientational ordering, so the molecules are all aligned in a way, but there's no periodicity. They're not fixed in a particular state. In a liquid, of course, the molecules move all over the place. So these liquid crystals have characters of both the solids and the liquids. They're state of matter formed by elongated rod-like molecules, in our case, spindle-like cells. And they have two main characteristics that allow us to say that a substance is a liquid crystal. So they have this long-range nematic orientational order, which is what you see here. And they have topological defects, which is what I'm going to show you in a second. So topological defects are characteristic of liquid crystals, and in two dimensions, they behave as particles with both charge and orientation. And I'm going to show you some real topological defects in a second. This one here is called the comet, and the cells align along the lines you see here. And this is called the trefoil, and the cells align. Here they move like this, here they move like that, and here they move like this. So why do you form these topological defects? Because you have cells that are aligned in this direction, and then they come in contact with cells that are aligned in this direction, and where they, in a way, hit into each other, they form these topological defects. And this here is from samples of topological defects of microtubule kinesin motor mixtures, which form very, very beautiful patterns of these topological defects. So why should we care about the topological defects? We should care because actually they have functions. These structures that are physical structures have function. They participate in the extrusion and apoptosis in MDCK epithelial monolayers. They determine the cell density and differentiation in primary cultures of neural progenitor cells. Very, very interesting data from the regeneration of Hydra, which is a little organism which you can cut in pieces and it will regenerate. Very particular topological defects appear where different appendages from the Hydra will develop. And also heart and liver have been shown to be organized as liquid crystals. So what can we say about the gliomas? Is there anything is there anything in the gliomas which allows us to say that maybe they actually have the structure of topology of liquid crystals? So the first thing I'm going to show you that there's long range nematic order of gliomas. You see this in the formation of these structures here, which we call oncostreams, which is these are fascicles of elongated spindle-like aligned and motile cells, and they form in different tumors. They also form in human tumors. And here I'm going to show you how they actually move. And you see, if you look along this blood vessel here, the cells are moving in two directions, going back and forth, exactly what you would expect of a nematic arrangement of the cells. And what you see here in green is the tumor cells, in red is the normal brain. These are slices that are put under a confocal microscope and imaged for 2 to 3 days. It's not moving. Can you move it forward? Great. So I'm going to summarize 7 to 8 years of work in the lab. These oncostreams are anatomically distinct. They're dynamic structures that move back and forth through different parts of the tumors. They contribute to the malignancy of the tumors. They contribute to invasion, and they contribute to cell spreading. So they have a role in setting up the heterogeneity of the brain tumors. We've shown that they are molecularly distinct. There are certain genes like genes that encode for collagen type 1 that are overexpressed in the oncostreams. And when we knock them out of the growing tumors, the tumors become much less malignant and actually lose the oncostreams. So I've shown you that we have the long range nematic orientation of the gliomas. Do we have topological defects in the glioma? So to do that, we actually went to in vitro cultures. So here I'm showing you the formation of topological defects in primary cultures of brain tumors. And what you'll see here, initially the cells are moving in any direction, and eventually they become like frozen or jammed. And if you look here, you see that the cells are aligned in a nematic fashion. They're all parallel to each other. But then here, there are very strange patterns forming. Here, there's strange pattern forming. And we had, we didn't know what was causing these. And here, this is a movie which is identical to the previous movie I showed you, but it shows you the orientation of the cells at each point. And what you will see, oops. Initially, the orientations go in any direction, but then they get, as the cells get jammed, they get fixed. And here you see a strange pattern forming. Here you see another, another pattern forming. And when you then superimpose the characterization of the defects, you see this is a trefoil defect, this is a comet, and this is another comet forming here. So what I've shown you now that we have evidence for both the nematic orientation of the cells and the existence of topological defects. $64,000 question is, okay, let's assume you have these topological defects. The organization of the tumors looks very much like an organization of liquid crystals. Do these topological defects do anything? And what I show you in this slide, taken from cells in culture, where apoptotic cells have been labeled with annexin 5, what you see here you see the accumulation, the orientation of cells. This is a comet, this is a trefoil. They're probably close to each other because they're going to fuse positive and negative topological defects and to fuse each other. And what you see here, both for the comet and for the trefoil, a higher accumulation of markers of apoptosis. So it could be The brain tumors are organized as liquid crystals, and that the topological defects have a role in creating these killing some of the tumor cells through apoptosis. Does this happen in vivo? Looking at the topological defects in vivo is incredibly complicated, and we are working with that with Carlos Blanc Mercader. But here you have an image which looks very much like a -1/2 topological defect or trefoil, and right in the center you have a number of marks of cells that were labeled with cleaved caspase 3 to look for apoptosis. So the conclusions, we postulate, we hypothesize that the gliomas are organized as liquid crystals. In support of this, I've shown you the existence of long-range nematic order, exemplified by what we describe as oncostreams. The topological defects are detected in vitro and potentially in vivo. And the translational implications are, we postulate that possibly disassembling this long-range orientational order, i.e. disassembling the oncostreams, and or deploying topological defects to induce apoptosis may be future ways of treating the brain tumors. So this is a slide to acknowledge we work together in a unit with Maria Castro, and the people who participated in this job, in this work I've shown here, is Andrea Comba, Ana Argento, and Faisal Sayed. So thank you very much, and I'll be happy to take any questions.
Hey, Pedro, how much is there an alignment between the underlying tissue architecture, like gray matter, white matter, et cetera, and these oncostreams?
Right. So there actually isn't, because we are looking at rodent tumors, and by the time they grow, any underlying tissue has been essentially eliminated. The structures where the tumor cells grow along white matter tracts or white matter, gray matter borders, you see that mostly in human tumors, because most of the tumor is preserved there.
That was so cool, Pedro. Really cool. Is it possible that the apoptotic cells are inducing the topological defects rather than the other way around? Because the cells that are dying can't participate in the oncostream anymore. And so they themselves would become a disruptor. So I'm just wondering, like, can it be modeled in that way?
Yeah, it could be. We really don't know who comes first. But it could be that the killing of the cells sets up a system where you form these defects. The defect, it's a complicated question, because the defect itself is an absence. It's called the singularity, and you detect it because there's actually no cells in there, or in our case, sometimes only dying cells. So yeah, it's a difficult question, but very interesting to ask.
Yeah, it'd be interesting to know whether the empty space is just a time point beyond the apoptosis.
Yeah, so we're trying to, what you're seeing is stills from movies, and we're trying to analyze those movies to see whether we can address some of those questions.
Yeah, it's fantastic.
Hi. It was really nice, great talk, especially inspiring too. And now I'm wondering, actually, since apoptosis is programmed cell death, did anybody have a chance to take a look at the embryological development and the apoptosis and regulation of their topological movement, and how it is like programmed, or is that programmed in a way?
Yeah, so the best information on development is really what people have done in Hydra, where they show that the whole structure of the actin cables underneath the epithelium in Hydra are forming a liquid crystal, and then they have defects where different appendages come. Elsewhere, no, it has been looked at cells in culture, but not in vivo.
Okay. Yeah, because for instance, even in mammalian systems, like hand formation, finger formation, or so, they also create first,
Yeah,
you know,
yeah,
and it looks like yours.
yeah,
work.
sure, but yeah,
yet.
but yeah, that's very interesting. What I forgot to say is that all of you have topological defects on yourself. Just look at your thumb, and you'll see topological defects of various types. And yeah, sure.
So in vitro, these cells are moving on the surface of the plastics. In vivo, you've shown an example of cells going against the vessel. Are there other ways, like in this last image with a group of apoptotic cells, what are they moving? So they need to grab on something to move. Are there any special extracellular matrix features, some like collagen fibers or something like this?
Yeah, the answer is yes, but we don't know what exactly it is. Collagen fibers play a role, and we've shown that in our previous work. In vitro, they're growing on glass with polylysin and laminin. What is exactly how they grow in vivo and what matrix they do is work in progress. But we're looking at what is the matrix that these cells create that they then move on. And collagen is certainly one of those proteins that participates in that.
Mm-hmm. So they essentially have kind of crawling motion. Do you have an idea of how they move? Because they're probably not swimming.
Yeah, I think it must be some sort of crawling motion, but exactly how it happens for the cells, we don't know.
This was fantastic, mind-blowing. You mentioned that this was a characteristic of normal tissues. It sounds like it's a characteristic of all biology. Is there any qualitative difference between the malignant liquid crystal formation and malignant tissue and normal tissue that we could take advantage of?
Probably, but very difficult to tell at this stage. The list I've shown you, or the list I showed you of the effects of topological defects in biology, that's essentially all there is. So it's really new. The paper on Hydra came out maybe a year or so ago, and there's a lot of information that is being generated at the time. We're collaborating with Carlos Blanc Mercader from Paris on trying to see these defects in 3D. And there's actually so far only one paper in the literature with a very nice but a very artificial system showing topological defects in 3D. So it's very much a work in progress.